Looks like I've cleared the back out pretty well, eh? Okay, green light's on. You know, where'd that come from? The ghost of Christmas past. Yeah, I'm checking on you guys. Alrighty. Just remind you of a few basic principles. This is carbohydrates this week. We do carbohydrates 2 next week, which means this week we're going down glycolysis, down that main central pathway. Next week we're going to go back up. But also next week we'll deal with PDH, which is just down the bottom here, going in the citric acid cycle. Some of your learned colleagues are rather nervous about how many metabolic pathways there are to cover. We're almost done. So next week, gluconeogenesis. The week after, remember you have Q&A tutorials. It'll be online. It'll be the same time as the lectures. That'll be on Blackboard Collaborate. You can go to any one of those times. So if you look on views, there's the times for Parramatta, which is the same time as you, 4 till 6 tomorrow evening. Hawkesbury has theirs 10 till 12. You can go into any session, totally up to you, multiple sessions, because Blackboard Collaborate doesn't have a numbers limit on it like Zoom does. They will be recorded. I'll take you through some key issues with regard to the report. You'll hear my dulcet tones. I'll show you a few things on Blackboard Collaborate, and then you have questions and you type them in. So you can hear me, I can't hear you, I'll see your questions. And when you type them in, they come up sequentially, and I'll basically paraphrase each question, just make sure I get it right, and then respond to the question, and then move on to the next one. Does that make sense? And again, you can go to any session, they'll be recorded. After that week, any questions on the report, it'll be NMP, and it'll be please watch the recordings, we've covered it. So any of your colleagues who decide to skip out. Now remember at the beginning of the semester I said that there were 12 quizzes, 10 of them count, yes? We had a practice quiz in the first week, that didn't count for anything, that was just to make sure you could do things, like follow instructions. But there is another quiz during the Q&A tutorials. But you don't have to be on campus, you can be wherever the hell you like, including McDonald's, stuffing your face with burgers and chips. Totally up to you where you are, it could be the gym. Is everyone clear on that? I'll put notices on view, so not next week. Next week we're doing carbohydrates 2, the week after. I'll put a notice on views to remind you. So that's one of your catch-up quizzes if you do better in that, so those will count. Also, this week you are dealing with Module 3. What are you bringing into the lab this week? Sorry? A manufactured or synthetic beverage. Please look carefully at the label, the nutrition label or nutrition information. If you can, try and get one with colour, but that's not critical. Some of your colleagues were asking questions previously to this lecture about what sort of things we deal with. What's the focus for this part of Module 3? Interference. In the lab, you're going to get BSA at a standard or stock concentration. You know what it is. Interference is about understanding the principles of what happens to try and measure your analyte. Now, if you look at a lot of these manufactured beverage, have you looked at them? Did you see how much protein was in them? 0. In theory, yes. Doesn't matter if it has a little bit. Does it have just protein, or does it have some other molecules that kind of look like some amino acid derivatives or some other junk they throw in there? Did you look to see how much sodium was in there? In the lab, you might want to keep in mind that you'll get some saline. What's saline? What's that whisper? What's saline? Salt, isn't it? What kind of salt? It'll be labelled saline, 0.9% WV. You have to work out what the concentration is. Now, if you're trying to work out interference, you know you've got a beverage that supposedly has no protein in it. Maybe it has a colour. Should you run the spectrum of that particular beverage? See what its maximum absorbance value is compared to your dye. Should you do that? Go like this. Okay. Yes. Should you look at your red dye on its own and with BSA? Go like this. Now I've got my beverage. I want to know if it interferes with the assay. What if I put some BSA of known concentration into my beverage? Not the whole bottle, don't waste the whole thing, just a sample, a known amount. And then I run my protein assay like you've been doing for the last couple of times with my red dye. If I put Y amount of BSA in, should I get the same amount back? Should I be able to measure the amount that I put in? Go like this. And if I don't, what does that tell me? Interference. Should I just use one concentration of BSA, or maybe 2, or maybe 3? Go like this. Wouldn't that be wise? Very simple procedure, but this is a way to check for interference, isn't it? Whether the red dye is affected by some things in whatever beverage you bring in. And what you're doing, and I want you to jot down the term, is spiking. Spiking the sample to see if you can recover. Now, if your beverage happens to have a small amount of protein, say it has X, and you add Y, how much should you get back in your assay? X plus Y, whatever that is. Totally up to you, isn't it? That's what you need to tell us when you finish with your samples next week. Should we get on with the lectures? Go like this. All right. We're starting with carbohydrates. We're just about done with all these crazy metabolic pathways. During the way, we've highlighted some key steps and also key regulators, key enzymes. And I've given you some models of enzymes, allosterics, covalent modification of them. We're going to see in this pathway there's a few very important enzymes that have different types of regulation. Why? Because this pathway is the carbohydrate from here down to here is glycolysis. You need to know the key steps in glycolysis. It's a core pathway. It tells you that from the schematic, doesn't it? It's a core pathway. Many things come into it and out of it. We've seen some of them. We've been skirting around this all evening, or the last couple of weeks, I should say. Next week, we're going to do gluconeogenesis. We're going to go back up, and you'll see it's almost exactly the same. A couple of bypass steps. And usually, note it down, bypassing a key regulator. That's what we have to do. Next week, we'll also touch on, right down the bottom here, the enzyme that takes the 3-carbon pyruvate into this citric acid cycle. That cycle there, I'm going to wait till after semester break. Put that into oxidative phosphorylation. Because remember, this is where you generate electrons and protons. And have to move stuff around in the mitochondrial matrix. Now, glucose is an amazing molecule. Yesterday, I had the privilege with some of my colleagues. We're isolating fluorophores from plants. We found an unusual one of my students, collaborative students, a glucose linked to a fluorescent molecule. There's a bunch of them. Stuff you eat all the time, like broccoli, cabbage, the brassicas. This fluorescent molecule we're particularly interested in because it's probably taken up by glucose transporters, and I'll touch on those today. Glucose diffuses through your membrane, but too slow to be effective for you. You have to get it in faster. Glucose is a nice, simple molecule. 6 carbons, but it generates energy. And the key thing I want you to write down, it loves water. That's why these plants use it, because it traps water for them. Why it's fluorescing, we don't know. It's what we're going to work out. We're going to see that once you get glucose into the cell, you can store it as glycogen. An individual glucose molecule loves water. As soon as you make a polymer of glucose, lots of branches of glucose together like a tree does, it's less of an issue for cells. It doesn't like as much water. That's storage. Now, glycogen, you want to jot down, occurs mostly in the liver and a little bit in muscle cells, I should say. Glucose running down here, oxidative via glycolysis, underline it to make pyruvate, so 6 carbons down to 3. But we might have to go back up because this is going to occur in hepatocytes. If I have a meal that's high in glucose, what do? Put it into cells. If I'm low on blood glucose, what does my liver have to do? Pump glucose back out again. I can also use glucose to make these things. I suspect you saw a lot of this with Tom Miller last semester. Glycoproteins, lots of branch sugars. Branch sugars give unique activity for molecules, particularly on immune cells and immune cell proteins. Lots of hydrogen bonding. Glycolipids, we saw those last week. Well, they give you your blood groups, A, B, and O, for example. Again, more branching. But too much branching can be dangerous. Too busy immune cells attacking you instead of defending you. We're not going to deal too much with the ribose 5-phosphate pathway. You can modify that, and we'll see a little bit next week where it's modified to help manage something like free radicals. I mentioned this diagram at the beginning of the semester. On the bottom of this diagram, one of the slides had KEGG, K-E-G-G. I suggested you go to that web page ready for your report. It's not the only place. I mean, everyone's going to go to Google, aren't they? But you need to go to some journal articles as well. I had a question before we started, and I'll go through a lot of this in the Q&A tutorials. How many references should you have for this report? What are the 4 headings you need? Research, diagnosis, treatment, and policy. You know, if I'm not seeing 4, 5, half a dozen references for each one of those sections, they'd better be pretty good, hadn't they? And I mean journal articles, not just web pages. And if you have a web page, you need a session date when you access that particular web page. Is that kind of clear? I don't have a maximum or minimum, but if there's minimum, and I'm a minimalist, it'd better be pretty good. But if you've got 40 or 50 journal articles, you want to think, well, are they really helping you talk about what you need to say? Can I move to the next one? Okay. We're going to deal with ingestion and digestion. You're taking glucose in to your blood circulation. We talked a lot about the process of putting food into your mouth. What happens in your mouth? You're masticating, increasing surface area. What's your saliva producing apart from steroids, cortisol? Amylase, what's that doing? Breaking up polysaccharides. An email from a student a little while ago. Oh, I've got a baby. I'm a bit worried about the smell on her breath. It's sort of baby's breath. It's rather sweet. Could that be the acetone we're going to talk about in diabetes later on the semester? Could be unusual, but it can happen. What about those polysaccharides? If I break them up into small sucrose and fructose molecules, what have I got? Sweet-smelling sugars. That's what babies tend to do, especially early in life. Need a chair, are you right? Wrong lecture. We're going to get those carbohydrates in, but we have to absorb them across our epithelial cells yet again. Once they get into the bloodstream, we've seen amino acids at week 4. What do you want to put in there? Lipids as well, but like amino acids, carbohydrates go into the blood circulation. So your blood glucose levels go up. In a few weeks' time, after semester break, you'll see that blood glucose going up actually stimulates insulin release. Insulin then tells cells to put more glucose transporters on the cell to suck glucose up. There's a whole cascade of events. In your islet cells and your pancreas, there's a whole sequence of using potassium and calcium ions to get insulin out. Then I've got this blood glucose in my circulation. Now this is in the fed state. Once it's in the circulation, I need to get it into a cell, often particularly the liver cell. But other cells will suck up glucose as well, particularly muscle, and your neuronal cells. Again, I've got to have a transporter. It will diffuse through, but too slowly. I've got to suck things in quickly. Cells are busy and demanding. Once I get it into the cell, my glucose molecule, because it diffuses in by itself, it means it'll diffuse out by itself, based on gradient. Once it's in, it has to be trapped. I want you to write that word down here. It's trapped here by putting a phosphate on it. If I stick a phosphate on it, it can't diffuse out again. With the phosphate on, what have I done? I've activated the molecule. What can I do with it? I can make a polymer like glycogen. You'll see that today. That's going to help save the cell from exploding with too much water coming in. Osmolarity. That glucose 6-phosphate means I can also run all the way down to pyruvate. Right next to that, glycolysis. And on the other side, put an arrow back up, a couple of dashes in between, gluconeogenesis. I can run down to glycolysis if I need energy, or back up in gluconeogenesis in the liver if I need glucose for the brain and other tissues. From the pyruvate, I can make acetyl-CoA right there, where that laser's zooming around. I want you to write PDH. Pyruvate dehydrogenase, we're going to see that next week. Massive molecule, we saw in that beautiful Harvard animation, big cup-shaped character, grabs the pyruvate and sends it off to the citric acid cycle. Now this is a core pathway. You need to know some of these key steps as we go, and I'll keep highlighting them. Are we done with this one? Yes? Okay. You've ingested. I've just sucked in a lot of, well, I had a hot chocolate a while ago to keep me going for a while. I won't be home till about 8 o'clock. Put in all those sugars in my mouth. I'm masticating that. There's the saliva, I've broken it up. I now have it in my stomach. What's happening to that hot chocolate in there? What's the acid doing to the amylase? Neutralised, just another protein, isn't it? What about your sugar, your carbohydrates? Not much going on now. Mushing around, okay, eventually I get it down. Forget the caffeine and the chocolate. Now I've got the sugars. They're running down to my intestines. Let's see what happens in the intestines. I run into the intestines, what do I need? More amylases. Where are they coming from? My pancreas. They're inactive enzymes. They're activated in your gut intestines. What are you working on? Well, these less simple sugars, well, the starches we're starting to break up. These amylases like maltase, dextranase and glucoamylases, they are often at the brush border, remember that? The villi, close to where you need to absorb those small glucose molecules before someone else gets them. So they're right there doing their job, absorbing them. Now, I mentioned a moment ago, as soon as you have that amylase from your saliva, like the baby's breath, going into their stomach, remember their stomach's not well formed, at least for the first few weeks and months. You're going to lose some of that activity of amylase. And even that protein becomes digestible protein for you as well, so you produce some of the stuff you're going to ingest. Then the pancreatic amylases get busy in your intestines. Doesn't take long. There's pretty busy enzymes. 5-10 minutes, they're already working very hard, and they're working at the brush border, trying to break up the more simple oligosaccharides into the very simple ones like sucrose, lactose, and fructose. Now, some of you know from different cultures that because of the effects of lactose and lactase, become deficient as you get older. Now, that doesn't change, well, it varies depending on your race, your background, and your exposure. Now, because in this country we tend to have a lot of milk, coffees, chocolates, whatever, you tend to be more tolerant of lactose because you've got it more through your life. It's training your cells to manage it. Some individuals quickly go off of lactating females, that is, they're not taking in milk. Quickly they have lactose intolerance. But you are training them because you're on a, bless you, on a milk-based diet most of the time. If you change your diet, you're changing your gene expression as well. So I can't stress enough. And that tends to happen for some people around age 4, unless you keep them on a milk-based diet. Can I move to the next one? Okay. Oops, that's not the next one. Now we're down in your intestinal lumen. Here's our epithelial cell, this big pink thing, those little fingers sticking out is the brush border. Now I need you to think carefully. I've just had that hot chocolate. I've got my brush border cell. There it is, my nice little villi. I need to absorb glucose in, but that's the external environment, isn't it? Yes? Even though it's the intestinal lumen, it's actually outside of my cells and tissues. I need to get glucose inside of me to be able to get into my blood circulation. To get it inside, I need you to look carefully at these slides. I need this green thing, that green thing, and that green thing. What are those green things? They are glucose transporters. This one is a sodium glucose transporter. That's an integral membrane protein that sucks up glucose. Before you look too far, right next to that, every glucose molecule it sucks up, I want you to write down 2 sodium ions. So for every glucose going in, 2 sodium ions go in from your gut lumen into your epithelial cells. And why do McDonald's love you so much? lots of sugar, lots of salt, and lots of fat. What do you do when you have lots of sugar? Take in lots of salt to your blood circulation. Is there a question, Prime Minister?
I was just going to say, if you don't have the sodium ions there, the glucose obviously can't transport.
It's harder to absorb, indeed, particularly for some individuals. So that green thing is an integral membrane protein. You'll notice it's often called a symporter. Sodium and glucose are going in together. 2 sodiums, 1 glucose. The other two, fructose and galactose, they're also taking in these molecules. Galactose needs sodium as well, and so does fructose. It doesn't need so much sodium. Remember, that's a 2 carbohydrate molecule in that context. A little bit easier. My epithelial cell might use some of those, depending on what stage it's at, how much energy it requires, but it's supposed to be a benevolent cell. It's supposed to give up those sugary molecules, glucose, fructose and galactose, into the blue stuff, which should be your red blood cell system, into the blood circulation via that glucose transporter. Now you'll notice that transporter, of sugar molecules particularly, is pumping sugar molecules out. No sodium comes out with it. To get sodium out, I need this brown character, which is an integral membrane protein as well. That's a sodium-potassium transporter. It's an antiporter. 1 sodium in, that's a positive charge out. Balance the charge, a potassium in. And your cells have to balance that, and that's an energy-demanding process. So that brown one, put next to it, ATP required. For all of these sugars down here, and this one pumping out here, no ATP required. They are termed passive transporters. They're pumping the stuff out because they're good at it. Please note the acronym, the sodium glucose transport protein, SGLT. Why is that? Because it's from the external environment into you. There's a whole bunch of glucose transporters coming at you that are actually in you, and they don't need sodium to get it into the cells in you. Is that kind of clear? To get glucose or fructose or any of these simple carbohydrates in, you need salt. We comfortable with this one? Yes? So think about that when you're down at McDonald's. Whacking the chicken salt on. There it is, lots of carbohydrates. What are you doing? Putting extra salt in your circulation, aren't you? Hypertension looking right at you. Should we move to the next one? One second. Yes, ma'am.
So you were saying something about the sodium transporter being from the environment. Which one was that?
From the environment. So over here, this is your gut lumen. So there's breakfast, lunch, there's a McDonald's burger running down there. That's outside of you. You're a worm, basically. That big long tube in you is actually outside. To get it from the tube into you, you need sodium to get through there. And just remember, it's 2 sodium ions. There'll be an exam question on that coming at you. Done now? Okay. Here's a glucose molecule. We touch on this a bit next semester. Have a nice little animation to show you. He says, hopefully, crossing his fingers that it works. Did before we started. Okay. Remember in FP and G, you talked about transcription, translation, and during this semester we've talked a lot about rough ER. Okay, there's a ribosome. 5', 3', what do you think that is? Sorry? Yep, which kind of bases? DNA or RNA? RNA, of course, it's on ribosomes, isn't it? So we're now going into the process of translation. What does your report have to talk about for your enzyme? The synthesis. It's made from a gene, isn't it? It's transcribed into RNA. It's translated into a protein. Is there post-translational modification? Is there multiple subunits? Is there disulfide bonding, methylation? Those are the synthesis of your enzyme. Is that clear for your report? That's what it's referring to, and I'll come back to that in the Q&As. This is making any protein. This little animation I did when I was doing the old Biochem 1 for a number of years. I'm about to make ER. That is the messenger RNA. This is the ribosome. Coming out here is the polypeptide. This is the protein you're about to make. That polypeptide has a signal sequence. Now, you've heard in messenger RNA, the start codon, the stop codon, the poly-A tail, all the other bits and pieces, maybe some isoforms. Well, in your polypeptide, that signal sequence says where that molecule's going to go, which might relate to your report. This signal sequence happens to be at the front end. Now, that signal sequence tells that polypeptide it's going to be made and going to go into the lumen of the ER. What are we about to make? Well, we're about to slow down the ribosomes effect. Grabbing this thing, this green character happens to be a signal recognition protein. It grabs that signal sequence of the polypeptide, slows down translation, and then finds this receptor. That receptor, trigger happy on the mouse today. That receptor is a polypeptide, multi-subunit. It's embedded in the membrane, like our glucose transporter we'll see in a moment. That's the signal recognition protein receptor. So it's looking for that green football. We had a bunch of kids playing rugby league at Hawkesbury all day today, and I mean a bunch of kids. Lots of football. Well, it's not really football, is it? Australian rules is the only real football. Okay, now this SRP latches on to the receptor. What have you just made? Ribosome ER, what have you just made? Rough ER. Now I'm going to continue translation. What I find is this thing, which is a translocon gate. This thing goes in and out of that membrane of the ER a number of times, has a little lid on it. This complex finds that thing. Maybe you missed a bit of text on there. This translocon gate finds it, opens up the gate. Beautiful biochemistry. You can open the gate. I'm just making rough ER. What do? I continue translation. I recycle these guys to find another polypeptide, to find some other football link to it. I'm going to continue translation, so this is rough ER. Continue translation. Now, often the signal sequence is removed from the polypeptide. It's done its job. Mr. Pac-Man comes along. There it goes. This is not Guardian of the Galaxy things. We continue translation. This could be your enzyme for the particular report you're dealing with. Goes into the ER, and often we start the process of post-translational modification. Folding, we might go through the Golgi, the cis-medial trans, other bits and pieces added, for example. We've now sent our molecule. This could be insulin, packaged in a vehicle and released later on from your islet cells. The same process. That's easy. Now what do I want to make? Well, in my liver cells, I want to make a glucose transporter. Now, a glucose transporter, you see over on the right, there's the lipid bilayer. It goes in and out of the lipid bilayer a number of times. There they are. How many times does it go in and out? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I don't have enough fingers. Some people might think I do. There's the fingers out of the membrane. They come around, look at my hands, and form a pore. That's a selective pore to grab glucose. That's how this glucose transporter works. Same with SGLT. But it's got to be embedded in the membrane, and those domains that helical through the lipid bilayer are mostly hydrophobic amino acids. I'm going to make this molecule using this kind of approach. There's my signal recognition polypeptide, looking for the signal sequence in red. I find my receptor. I find my translocon gate. I open it up. I continue translation. I recycle those guys. Continue translation. There's no Mr. Pac-Man this time. Remember, those amino acids are likely to be hydrophobic. I don't need to clip them off. I end up with some of this polypeptide out of that membrane and some more inner the membrane, depending on how I set it up. Now, this translocon gate is like the glucose receptor. It opens up to allow the protein through. There it is, allowing it through, forming that helical domain through that particular lipid bilayer. That's the same phenomenon you see up here, except it's gone in and out a dozen times. Does that make sense? That's how you've made an integral membrane protein. Let's go back a couple of slides. This character here, you've made those things the same way, except you moved that bit of membrane from the ER through the Golgi out to the cytoplasmic membrane and fuse it. It's stuck there. Eventually you'll recycle it. But I need you to know, particularly on this slide, not this slide, not that slide, oop, that slide, dizzy, too much chocolate. This is my glucose transporter. There we are, the dozen fingers going through the lipid bilayer. Look at my fingers. I've just formed a pore. I'm now looking at the pore directly at you. That pore now I can rotate and ratchet around, which is what you see here in this schematic. These amino acids here, particularly asparagine, serine and threonine, form that centre ring to be able to grab glucose and suck it into the cell. If it was SGL2, it would grab 2 sodiums too, wouldn't it? It's sucking that glucose in via that pore structure. It's a selective molecular pore in the membrane. So there we are sucking glucose in, but how do you control how much of this molecule's on the surface? Well, that depends on what you eat. Let's have a look at this schematic down here. Finished writing? Not yet. I heard you. I love to disagree, but not for the sake of it.
Does it have 12 translocal gates?
Sorry?
Does it have 12 translocal gates or only one?
Question was, does it have 12 local gates? No, if you can imagine. Can you put up 2 fingers, please? 2 fingers. Up high, come on. So they are the 12 membrane proteins in and out of the membrane, okay? We are going to form a pore like that. The inner rings are asparagine, serine, threonine inside that ring. We're going to grab glucose in that ring. It's only one pore. Does that make sense? Okay. Thank you for the fingers. Now, please note passive transport. Write down no ATP, no GTP, no GDP, no energy required. But also note that it increases the rate of glucose uptake 50,000-fold. That glucose molecule we isolated from plants the other day, I put onto some cells and it's fluorescing inside the cells within 5 minutes, probably through glucose transporters. So what you do is put more of these on the surface after you have a meal. So that hot chocolate I had, it increased my insulin levels, and my insulin levels told cells to put more glucose transporters on the surface. We're ready to look at the part on the left now. Gentlemen? That's a thumbs up, that's good. Here is a cell. That's the lipid bilayer. Here is the insulin receptor. You're going to see more of it as the semester progresses. Involved obviously in diabetes and a few other issues. There's insulin. A, B form, disulfide bonds, made the same way we saw a moment ago, pushed out of the cell after a meal as glucose levels rise. What does it do? It sticks to its receptor and signals. You'll see G proteins again, a bit more detail as we go on. It signals to this cell. See these glucose transporters here? They look like a clothes peg, don't they? So there's my clothes peg. Grab glucose, grab glucose, grab glucose. Inside you, is there any sodium required? Go like this. No sodium required. I've just had a meal. If I have to go back to transcription, translation, post-translation modified, stick it in a membrane, shovel it out to the cell, it's going to take hours and days, isn't it? Wouldn't it be easier if I could have these glucose transporters in a little vesicle membrane, and every time I have a meal, all I do is tell the cell, put it on the surface. And when my blood glucose levels drop, glucagon comes along and says, there's not much blood glucose, don't suck it up, send it out. What should I do? Put it back inside the cell. If it's inside the cell, it can't suck up glucose, can it? If it's on the cell surface, it'll suck up whatever glucose is in your blood circulation. Write that down on the slide. So what you see in this schematic is insulin telling the cell to fuse that lipid bilayer, that vesicle with glucose transporters, put them on the cell surface. The blue dots are glucose, and suck up as much glucose as you can in your liver, in your muscle, and store it as glycogen. If it's in its liver and you've filled up your glycogen, you're going to make fat, aren't you? So what you've done is put more glucose transporters on the surface. When your blood glucose declines, you put them back into vesicles, put them inside the cell. Every few hours, you're putting glucose on the surface of cells and then bringing them in as your blood glucose drops. Does that make sense? Critical exam question. That's often the GLUT4 transporter. There's lots of family members of these glucose transporters that are cell and tissue specific for a reason, because the liver's got to do the job of taking up and releasing. This process is called translocation, moving this glucose transporters from inside the cell to the cell surface. Can I move to the next one? You guys right? You guys okay? Yep, okay. All right, here's some family members you need to get to know. We're going to have a family reunion at the end of the semester. It's called an exam. GLUT1 on all of your cells. It has a Km. Now, what's a Km? I want you to jot down, it's a binding affinity maximum. How much glucose it can grab. That's not a lot, 1 millimolar. Takes up basal glucose. GLUT2, I want you to underline liver and pancreatic beta cells. Underline liver. GLUT2, be an exam question. The pancreatic beta cells, what do they secrete? It's not glucagon, it's not CCK, it's not somatostatin. What is it? Insulin, write it down. Insulin is going to come out of those beta cells. That has about 15 to 20 millimolar. It's a bit busier. I'm not going to test you on KMs or KAs and KDs. That's for next semester. That's dissociation and affinity constants. You're dealing with this guy because this guy is going to manage blood glucose coming and going. GLUT3, you also see on tissues as well, 1 millimolar. GLUT4, underlying muscle, underlying fat. GLUT5 we see in the small intestines. They need glucose too. And where do we see GLUT1s, particularly in erythrocytes? Why in erythrocytes? Why your red blood cells? What's missing in them? And nucleus is missing, and mitochondria. They rely on glycolysis to survive those few months. GLUT2, high Km value, particularly in your liver cells. Known to line the process, never saturated. If there's glucose, they're going to grab it. That's their role. Others, I'm going to slow down. If there's too much glucose, I've done my job. But please note this bit I've underlined for you. I should put italics and bold to make sure it's clear. Make sure you put a circle around it. Those same glucose transporters involved with putting glucose back out of a liver cell. So it's a two-way system. GLUT3, we see that mostly in your brain. So hopefully you have lots of GLUT3 transporters in your neuronal cells doing their work. GLUT4, we see those in the vesicles for insulin, moving them to the surface of our beta cells to release them out. And GLUT5 we see in the testis, kidney and sperm. Well, sperm use fructose as the major glucose supply of energy. Sperm have to mature in what we call immunologically privileged site, which is the testis. You need to know these. What's the difference between these and the SGLT? Sodium. Is there any sodium on these guys? Write down no sodium. That'll bite you later on. We done with this one? we there yet? Only SGLT. Why is that? Because you're going from the external environment in you. Remember, all of these are in you. They're in cells and tissues in your blood circulation. Okay? Can I move on now? Okay. Now I've got glucose in a cell, and I told you before, glucose on its own loves water. It will attract it into the cell. As soon as the concentration of glucose gets high enough, it attracts so much water, you can actually explode your cell. What does your cell do, particularly your liver and muscle? Well, anything over 0.4 molar is going to be dangerous. What do your cells do? Well, they take this individual glucose molecule, they activate it, we'll see shortly, and they form glycogen. They make a polymer. You need to know the enzyme I've circled in red, glycogen synthase. It's phosphorylated to inactivate it. What does that mean? Covalent modification, phosphorylation to turn it off. You end up forming these polymers which form these branches you see over here. So this green purpley stuff here is a polymer of glucose. It's all highly branched. Now, if this is in your liver and your muscle, particularly your liver, and suddenly you stop eating and you're low in blood glucose, and you need to think in an exam or a lecture theatre, well, maybe don't think in a lecture theatre, but maybe the exam, before you hit the panic button. What is your liver being told to do? Release glucose. Before you go to the exam, have a couple of lollies, you know, those natural ones, just to get your blood glucose up. Stay away from the caffeine. I've had to deal with students who have too much caffeine and go into almost cardiac arrest. I'm serious. Don't do that. But increase your blood glucose, ready for the exam. That's what athletes do. They use a lot of carbohydrates, don't they? A lot of pasta and so on, to get them ready for the next day or so. That's my polymer of glycogen there, polymer of glucose. Why is it so branched? Why isn't it kind of long linear structures? What's the advantage of being branched? Particularly when I'm in low blood glucose. Think about it. Chris is. Exactly. If I had some long linear thing and I needed blood glucose like a long polypeptide, I'd have to cut it up in fragments and do all the other bit. But if I've got heavily branched, I can clip off branches off the end, can't I? A whole lot very quickly. Glucose goes out very quickly. So my accessibility to that glucose is because of this polymerization effect. Here you see a nice little micrograph of glycogen granules in muscle and liver. They're little storage vessels, like an ATM. I can get it in and out quickly, unlike going to the bank. The trouble is, to form that polymer, I need a seed. This seed is called G, or glycogenin. It's basically a little polypeptide that has a tyrosine in position 194 that helps seed the process. I've primed it, ready to form the polymer. Now, often keeps the polymer, makes it branch out a lot when lots of glucose is available, and come back to not much. But branch, come back, based on a meal. We're done with this one? All right, one small thing. This computer's grabbed A, that should be alpha. Remember in FP and G, you had the alpha 1,6 linkage. If you have the beta 1,6, you might be a tree. Can I move to the next one? Sorry?
Oh, yeah, I was just laughing at the tree.
Oh, the tree, yeah. It's the same glucose molecule. We move on? Great. Now we have a concept to deal with. This is an enzyme. It's going to deal with the process of managing glucose. Who is it? I'm going to step you through it. It's glycogen synthase. What does this molecule do? Synthesizes the polymer. Grabs your glucose, here it is, glycogen synthase. When would you make it and how would you control it? Well, glycogen synthase is controlled by this enzyme, GSK3, not GlaxoSmithKline, not Pfizer. That's a kinase. What does a kinase do? Phosphorylate somebody. When should you be dealing with this? Well, if I've just had a meal, what should I be doing? eventually storing lots of glycogen. If I'm fasting, what should I be doing? Releasing glycogen. So you have to control this enzyme. Here we are. This enzyme's going to be controlled. If it's active, right next to that, it's making glycogen polymers. That's in the fed state, right? Yep, write it down, the fed state over here. It's active. See lots of hydroxyls hanging off of this little protein as well. To then get it ready to inactivate it, what do I need? Casein kinase, or CK2. What do I need? ATP. ATP's coming in. Here comes more ATP with GSK3. What do I do with GSK3? Phosphorylate it. Now it's inactive. Covalent modification to inactivate this enzyme. It needs to be clear, particularly to your liver, but also your muscle. Who says to this GSK3, think of GlaxoSmithKline, when should you not be inactivating this particular enzyme? After a meal. What happens after a meal? Your insulin levels are up, aren't they? So you should be inhibiting that process, hence the red cross. Here I have now my inactive enzyme. Who activates it to get busy again? This PP1, I want you to write down protein phosphatase 1. Protein phosphatase 1. Phosphatase tells you what? Taking phosphates off. Who's had the phosphates ripped off? Glycogen synthase. Now it's active again. There you go, the phosphates. Remember, we had to have 3 of them on there, so it's a lot of phosphorylation. In this particular case, who stimulates that process? Well, wouldn't you think insulin? Just had a meal. Get busy, store glycogen. Who else stimulates? Glucose 6-phosphate. Let's go back a couple of slides. Where do we see that character? Isn't that the active form of the glucose trapped in the cell now? It says get busy, store glycogen. Who else? Glucose. Well, if I've got lots of glucose, shouldn't I get busy storing glycogen? Yes, that's what you should do, until your glycogen stores are full. What about glucagon and adrenaline? Why would they stop the process of removing the phosphates, making that thing active again? What's the converse? Can't hear. Because you need glucose. So should you be storing it as glycogen? Heck no. So stop the enzyme. Don't let it reactivate. Don't let the reaction to form glycogen to take place. Please note this is covalent modification of enzyme activity. A simple enzyme making glycogen. The next bit is kind of important, because this bit we're seeing your insulin via its receptor, which looks a little bit like, kind of a muscly guy lifting a barbell thing, like me. Receptor stimulates it. That's what those little dash lines mean. We'll talk about signal transduction a bit after the break. What does that do? It stimulates more glucose transporters on the surface. Which cell are we talking about? Muscle cell. What does it also stimulate? Trapping glucose. I want you to write that term down. Trapping glucose in the cell. Who traps the glucose? Hexokinase. Puts a phosphate on glucose to trap it in the cell. It can't diffuse out now. It's going to run down parts of glycolysis all the way down to here. We can use some of it to go into forming glycogen with this glycogen synthase. What does insulin do? Stimulates that as well. Is this the fed state over here on the left or the fasted state? Fed, write it down. Exam question. The short answer will talk you about asking you to tell me what happens in the fed state. Sorry? Sorry, what? This is the fed state, isn't it? If you've got lots of blood glucose, you're telling cells store glycogen, store fat. Okay? You done with that bit? Okay. So in this case, insulin's job is to trap glucose. Trap glucose in cells, not just put more glucose transporters on, but keep it in the cell and stimulate glycogen storage while you've just had a meal. Think fed state. Then it gets trickier. What about the counter-regulatory hormones, adrenaline and glucagon? What's glucagon? Fed, fasted or stress state? Fasted, write it down. What's adrenaline? Fed, fasted or stress state? C, stress state, particularly acute stress. What do those characters do? They stick to their receptor. I'll talk more about their receptors later on. GS alpha, didn't I introduce that to you last week? Write down G proteins. Last week I said there was a signal transduction and it generated cyclic AMP. Didn't I ask you to write down capital C AMP? Or little c AMP, doesn't matter. There's cyclic AMP. What did it activate? Lots of protein kinases. There they are, protein kinases, a whole lot of them. What's this doing? This is amplification. Write it down. You're amplifying the signal. I stimulate 1, that stimulates 2, that stimulates 4, that stimulates so on and so forth. You're amplifying or magnifying the signal. More and more enzymes to do the work as quickly as possible. So you're recruiting in there, and that's what you're seeing, more and more molecules recruiting in there. In comes calcium. You'll see how important that is. You can't get away from calcium. It's involved in muscle contraction, cell signaling, even egg fertilization to shut down the membrane so no other sperm can get in. It's stimulating this process to turn all these characters on. Increase AMP, what's that? Adenosine monophosphate. It means my cell doesn't have much energy. Enough to go back and make more energy. Currency is ATP. That's all the steps we're running through for these characters to say, well, what am I going to do with this glucose? I can store it as glycogen, because remember, this is the fed state. But if you're in the fasted or stress state, what do you need? Blood glucose to work or run away, whatever the case is. Time for a break? No, you don't want to have a break? Portia, do you want to just nudge him and let him know there's a break time now? Oh, totally. Okay. I know you want to get back to work because you're all busy being noisy. We're going to deal with glycolysis. I've kind of stepped you through how you get the glucose into a cell, how you trap it there, how you form glycogen. Now we have to deal with the grunt pathway. Now, this is basically an overview to give you an idea that we've dealt with proteins. What did you do? Use them to make various things. Often you end up with acetyl-CoA. We dealt with fats. What do? You do beta oxidation. What do you end up with? Acetyl-CoA. Now, glycolysis. It's one of those TV shows, the Samurai Warriors, or whatever they call it, obstacle course thing. The athletes, that's right. They look like athletes. What did you make? Acetyl-CoA. In that, you could go into the mitochondrial matrix. Glycolysis is not in the mitochondrial matrix, it's in the cytoplasm. That's why I keep harping on red blood cells. Mitochondria disappear, you still have glycolysis. You're going to see next week that that's kind of important because tumour cells will use glycolysis to ramp up all of the enzymes we're dealing with today to try and get themselves a lot more energy to become a full-blown tumour cell. And we can use that in various ways to treat tumours. We're seeing glycolysis. I want you to write it down here to form acetyl-CoA. We're going to run through pyruvate. Once we've got acetyl-CoA, we can go into this yellow box. And I want you to write there, mitochondrial matrix. That's where we go in to make more energy through the citric acid cycle you see there. Again, that's after the break. And then we'll see oxidative phosphorylation after the break. Now, I keep pointing out this is a metabolism unit. I'm not a fan of microbiology. I hate microbes in my cells particularly, so I hate talking about them. But be aware other units talk about the nature of glycolysis in different species of various kinds. Let's just touch on a couple. Glucose, a number of steps down to pyruvate, underlying glycolysis. With an arrow back up so you know, a couple of dashes, gluconeogenesis. That's next week. That pyruvate molecules under anaerobic conditions we saw earlier on the semester, you form lactate. What do you do with that? Busy working muscle, you send alanine back out. That can damage the muscle tissues over short periods, but also microbes can use that. If you're so inclined, you might make some happy juice, ethanol. Ethanol. That's fermentation. I'll let the microbiologist deal with that, particularly down here with the yeast. Few are going to go through aerobic conditions. Now you'll note through glycolysis that we don't use oxygen. We only use oxygen down here in oxidative phosphorylation. You used a little bit in some of that beta oxidation process before. You'll run into the citric acid cycle from a 6-carbon glucose. You make 2 3-carbon pyruvates. You run down to acetyl-CoA, you've got 2 acetyl functions then, you're going to lose a couple of carbons of CO2, so you're losing lots of CO2. Down the end of the citric acid cycle, you clip off for a few more carbons and you produce water. In here, I want you to write, this is where you use oxygen. The last step, complex 4 protein. In the ETC set, I've already introduced you to. Right. You need to know this pathway well. This is a core pathway. Today I've just touched on some key regulators, talked about various things, danced around bits and pieces to give you an idea of simple amino acids, simple fats. You need to know this. Glycolysis is split up into 3 major components. Stage 1, trapping the glucose. Who traps it? Hexokinase. It needs a phosphate to do it as ATP. If you don't have enough ATP, you can't trap glucose in the cell. Glucose 6-phosphate, didn't we see that at the beginning of today? G6P. Glucose 6-phosphate is going to run down through this thing.
this isomerase, I want you to notice as we go down the one-way and the two-way arrows. Every point you see a one-way arrow, we have to go bypass it because we're going back up gluconeogenesis. If it's a two-way arrow, it's part of gluconeogenesis. So once we know glycolysis, we essentially know gluconeogenesis. We're going to run down to this next step to form fructose 6-phosphate. There's fructose. This character, I want you to write down PFK. I want you to write down PFK1, phosphofructokinase. What does it do? It grabs another ATP and makes F16BP. I want you to write down F16BP. Note the 1,6. Because that will bite you as well, where it hurts. I've got that. What have I got? Look over on the right. I've got a sugar molecule with 2 phosphates hanging off either end. It's really active. I'm about to split it in half, essentially. That's the next stage, stage 2, where we split the 6-carbon, which is now fructose, into 2 3-carbons. You'll notice they're interconvertible. Glyceraldehyde 3-phosphate. Dihydroxyphosphate. Didn't we see glyceraldehyde phosphate a few weeks ago? Yes, we did. Isn't that a simple 3-carbon molecule, a phosphate? We can do various things to it. We can take it into glycolysis. From that, I can intervert with the isomerase. I can run down to the next phase, also 2-way arrow. Here I grab NAD plus and generate NADH. Note that circle. You just generate electrons and protons in the cytoplasm of the cell. I run down this thing, phosphoglycerate kinase. What have I got? ADP. I've just made ATP. Here's your red blood cell making ATP. Hang on a sec. Stage 1, didn't we have to use a couple of ATPs? So, you know, your cost benefit's not working too well just yet, is it? Right. I run down from this kinase down to this mutase to form this thing, 3-phosphoglycerate. We've seen that character before. With enolase, there goes the water disappearing, and there's our friend PEP. Write down PEP next to phosphoenolpyruvate. PEP is the last step before we go through this enzyme. Underline pyruvate kinase. What did pyruvate kinase do? Well, it made pyruvate, but it used ADP, and what's its product? ATP. Now, hang on a sec. Take a glucose molecule, 6 carbons. They had to use a couple of ATs to split it, ATPs. Now I've just generated a couple of ATPs, so my net balance is not quite 6 carbons to 2 3-carbons. What do I have to do in this second half? Multiply by 2. So when a red blood cell, you use 2 ATPs to activate a sugar to cut it in half, and how much did you generate overall? 4. Subtract the 2 you put in, how much did you end up with? 2. So your efficiency in your red blood cell is not great, is it? It's enough to keep alive for a while. I want you to write in the top half, that's the input phase. The middle half is the breaking up phase. You know, I'm broken up with you, sorry, I won't see you again. But, not you again. Sorry? Not quite that much. We were friends. The bottom stage, I want you to write the payoff stage. Payoff. This is when you get your ATP payoff. But let's look at PEP down here. It's important for you. Have you looked at that assay I put up on views on Friday? What's the next real assay? Real assay? The one I put up on views, the suggestion you need for the lab this week and next week is? Oh boy. Oh boy. The real assay is called the PEP assay. Ah. Well, if you have a look look at that, what I'm about to talk about is not going to make a lot of sense, is it? Being prepared for metabolism in Mark Jones, as in Mark Jones, would be kind of useful, wouldn't it? PEP assay, your will assay. PEP. It's trying to measure PEP in a sample. I'm going to give you some tissue sample. You have to measure PEP. How do you measure it? If you look at that will assay, it does an enzymic conversion. Which enzyme do you think it might use to convert it to something you could measure? Write it down. Pyruvate kinase. What does it produce? ATP of pyruvate. That's the will assay. And why is that important? Well, the assays you've done so far, the lipid one measured what? The acid function. What about the one before that? What did that measure? The alanine assay, what did that measure? This current assay uses an enzymic conversion. So you're actually measuring PEP. You're looking at the efficiency of the enzyme, the pyruvate kinase, to convert it to something you can measure. Does that make sense? I suggest strongly you look at that will assay before you go to the lab, particularly for the workshop, because it's a conversion assay. An enzymic conversion, it's not a direct probe. Keep that in mind for your analysis. And you'll have to convert back to weight. Each will assay increases complexity just slightly to make you think about what's going on in the assay. So down here now, we'll write down PEP, put a circle and arrow for the will assay, PEP. I'm using a conversion assay. And an enzyme requires what? FP&G. Temperature and time, doesn't it? How much substrate? Clearance rate. Enzyme efficiency, EC number. All of those are critical for how well that enzyme works to convert PEP to its products for you to measure, which might have an impact on your assay efficiency, might it? And why does it relate to interference? We're doing interference this week, aren't you, in the lab? So you've got a kinase making something. Does something interfere with that enzyme? Yeah. Salt of different types, temperature, too much substrate, other factors that might look like the substrate but not the substrate, too much product build-up, all those things, too much time, not enough time, can interfere with the assay. That's why it's related to the interference component we're dealing with now. We're done with this one. Everything is related and very deliberate. Now let me go back one slide. Up here you see hexakinase. What did you do with hexakinase? You trap glucose in the cell. What would you trap glucose in the cell? In the fed state. Yeah. Now remember, this can happen just like that. I've just had my hot chocolate, now I'm in the fed state, aren't I? An hour or two from now, I'm going to be in the fasted state, driving home. What we call it. I've got to trap glucose and then I've got to let it go again. What do I need to do? Manage this enzyme differently. We've seen, I want you to note, we've seen allosterics, relaxed, busy, stressed, covalent modification, alter them, simple enzyme connections for last semester, ways to modify the enzyme. Today you're seeing a totally different thing. You're going to see this enzyme parked in the garage, the nucleus, told to come out and do its job, trap glucose. When there's not much glucose, park it back in the nucleus. Does that make sense? Hexakinase. That's what you're looking at on the slide. There's hexakinase. Here it is out of the cytoplasm of the cell. Who stimulates it to come out? Lots of glucose. What do? Tell the cell, trap it. Keep it in there so you can either put it in storage or use it. There's my glucose transporters. There I am running through a form like trap glucose and whatever I do next. Now, if glucose 6-phosphate builds up, I want you to put a square bracket and a little arrow up to it. Remember the cars building up on the lot? If they're not being sold, what do you tell the manufacturer? Stop making cars. Well, this one says when I build up that amount of material, I'm not using it for the rest of glycolysis. There it is up here. If I'm not using it, it's building up. What do? Tell that enzyme to park back in the garage. Does that make sense? So instead of inactivating the phosphate or activating the phosphate or making it relaxed or tense, put it away. Bring it out. Put it away, bring it out. Just like that. No back to transcription, translation. No messing around with some other enzyme. Just put it away, bring it back. When you bring it out, when there's lots of fuel. When you put it away, when you've got no fuel. Same with your car, right? No car fuel, back in the garage. Get some money, get some fuel, go for a drive. That's how this thing works in your cells. There's hexakinase, that's why it's mostly a one-way arrow, but it can go back a little bit as well. And where would it go back? We're going to see a little bit in the liver. Why? Because it's got to pump glucose back out very quickly. Many of these are cell or tissue specific. This is hexakinase 4. Hexakinase 1 to 3 different family members, we see particularly in muscle, is inhibited by G6P. What does that mean? Yeah, why? Why would your muscle be different to your liver? Because of the activity. I'm busy working. I need it, so I need lots of glucose 6-phosphate. That's the key point in this. It has a high affinity. That's the Km value here. So the numbers don't make sense. Hang in there and look at KD values. You'll see what I mean. And what you're doing is balancing uptake and workload on a second-by-second basis, based on whether you need to work now, run away, depending on what the case is. Move to the next one. Yeah. Have you noticed when that car is parked in the garage and the nucleus is bound to something? That character's a military protein. Grab it in that nucleus so it doesn't do anything else except just sit there. Until glucose goes up, let it go back out. Like an electric car. What's that dot point mean? Liver hexakinase 4, aka glucokinase, is not inhibited by G6P. Why is your liver different? We just talked about muscle and go, they're busy working. What is the liver supposed to do with excess glucose? If glycogen stores are full, what does it do with extra glucose? Turns it into fat. You don't want to stop doing the process of trapping. The liver's job is to make fat from that excess glucose. Good thing is you start to get the idea. It's actually really simple if you think the same kind of way. How your cells are working. Move on now. Okay. Here's the first phase. Remember I said PFK1 was a key enzyme. Let's just run through briefly this slide. There's glucose, there's my hexakinase, family members 1, 2, 3 and 4, depending on which tissue. Uses ATP to put the phosphate on glucose, but you'll notice a couple of cofactors. I keep hinting at things like in your juice, beverage. It's got lots of salts like magnesium, manganese, and particularly those two, manganese, we see in gene transcription, involved in some of those enzymes. They're cofactors to make them work, so if you're deficient in those, your efficacy of making this trapped glucose drops off. I run down this isomerase, I've made fructose 6-phosphate. This key enzyme I've highlighted in green, PFK1, is a key regulator of glycolysis. It makes, please underline again, F16BP. We are looking at the first phase where we're inputting. A couple of little crazy animations we can watch. Glucose. This one runs a bit quick. The input phase, 2 ATPs, 6 carbon molecule. you end up with 2 3 carbons. I'll trap somebody in the exam, I know I will. The output phase. You produced a couple of extra ATPs, didn't you? Yes? Put 2 in, you got 4 out, so that means you got a couple extra. But what else went in and out? NAD+, what did you produce? NADH, electrons and protons in the cytoplasm. You're not going to go into oxidative phosphorylation or electron transport chain. That's for the cytoplasm. But it's also, I want you to know, potentially for the nucleus. The nucleus uses NADH to go into the first events of gene expression, particularly in stem cell differentiation, usually in the first 3 or 4 hours. But that means electrons and protons come out, so not just ATP, keep that in mind. Again, I'll trip you up in the exam. What's the overall payoff for glycolysis? PFK1. Can I move to the next one? Yes. PFK1, last step before you split. You've got F16, 1,6, carbon 1, carbon 6, phosphate on each of them. Basically, this thing is busy on either end, ready to split half. PEP I've already mentioned, that character is part of your will assay. That's the enzymic conversion, pyruvate kinase, ADP to make ATP. What if you don't have enough ADP in your will assay? You can't make a conversion, and that has to be in excess. The payoff phase down here now after we split. Let's look at this character quickly. You'll notice it's 2 times over here I put, because it's 1 glucose molecule. You can stop there for a moment. Please note, as this is running just off to the side, and I'll just make this a little bit smaller. The NADH is coming out, 2 of them. The ATPs, 2 of them. And these are the steps. Now, before I take you through the steps completely, how many one-way arrows do you see in this particular part of the payoff phase? Only 1, which is your will assay, isn't it? That means gluconeogenesis can use all of those enzymes to go back up in the liver cell. So I only have to memorise 1 part of the pathway, don't I? But I know if I want to go back up, I've got to bypass this one, which we'll see next week. Let's have a look at the steps here. What are you doing? It's simply putting phosphates on somebody to activate them. Now, the critical thing about glycolysis, which is different in the mitochondria, see the isomerase moving the phosphates around. Let's get to the next one. In comes our inorganic phosphate and NAD+. What did you do? You stuck another phosphate on this 3-carbon molecule, 1,3-bisphosphate. Out comes NADH, highly charged electron-proton carrier, ready to do some work as a cofactor for somebody else. What happens next? I think I'm going to be an old man before it happens. ADP comes in with the kinase. Now, what I need you to jot down, this is enzymic production of ATP. You're using glycolysis, you're using kinases to make ATP. What is your substrate? ADP. What's your product? ATP. This is different from that rotating ATP synthase we saw in the mitochondria. That is not truly enzymic. That is a vice. It squeezes the 2 together. This is a catalytic reaction with a kinase. ATP synthase is not a kinase. Does that make sense? It works differently. Squeezes the 2 together, overcomes the energy, forces the formation of ATP. This thing is moving phosphate around. It takes them from this BP difference between glycolysis and oxidative phosphorylation. Let's watch the last few steps. So what do? I charge up a carbon molecule, I bring in NADH, and I generate an ATP. I do that through most of this pathway. I get rid of the water. What do I do now? Bring in another ADP. I use a kinase after I kick that water out. There's my friend PEP. There's my phosphate, highly charged between those double bonds and the acid function. Bring in ADP. What do I make? ATP. Enzymic production of ATP in the cytosol. That's what your red blood cells have to do all the time. That's why you don't need mitochondria in those cells, but their efficiency is low, isn't it? What's their workload? Drag oxygen carried around, convert CO2 out there, back in the lungs. That's all it does. That's why it doesn't last very long. But also, if you're a forensic scientist and you're looking at just red blood cells and you want to get DNA, you're going to give it in? No, there's no mitochondria, no nucleus. So the forensic people got tripped up on some of that stuff. They're looking at immune cells particularly, which have different rates of turnover to telomeres. You going with this one? Now, PFK1, here it is. It's an allosteric enzyme. You'll note yellow, green, brown, and blue. Multi-subunit enzyme for catalytic cells. If it's allosteric, think relaxed, busy, hence not so busy. How does this particular character work? Well, when it's relaxed, that's what the R stands for. It starts to bind F6P. Let's go back here. What is this? I'm going to run through this pathway. Let's go back here. F6P. PFK1 to make F16P. Let's go down here. High ATP acts by forcing the 10th state. What does that mean? If my cell has lots of ATP, what am I telling my cell to make? More ATP? No. I can slow that down. Put in my red blood cell. So lots of ATP inhibits the enzyme but doesn't stop it. It just makes it tense, slows it down. High AMP binds to the relaxed state and overcomes the inhibitory effects. What does that mean? I have lots of AMP in my cell. What is my cell going through? It's like here when you're running a cash in a tube, isn't it? What do you have to do? Get a job, get some more currency. So the same deal. You're going to get a job, make some more ATP, because I've got too much AMP, too much there. No? It's all the same deal. Our PFK1 has two binding sites for ATP, a substrate site and an inhibitor site. Substrate site and inhibitor site. Substrate, I can manage it, let's go back a slide or two. Substrate, there's my ATP, and outside of that, an inhibitor site. Not in the active site. Like I'm busy working, someone else is giving me a tap on the shoulder to slow down. But I'm not going to alter what my active is. Oops. Okay. The substrate site binds to ATP in the relaxed and tense state. The inhibitor site only binds the tense state. Think about it. Busy working, someone says slow down, okay, I'll slow down. My first job working university student driving trucks at a winery, working too fast. Don't work so fast, students tend to work hard, try and get a good job, keep the job. The regulars, slow down. If we work too fast, we'll lose our jobs and we'll have nothing to do. Look out the window a lot. What you want to do is slow it down so it keeps working but doesn't stop. Because the way your cells all need ATP at some point, and then sometimes you have to ramp it up and then slow it down. Depending on whether you're fed or fasting. What about this thing? Inhibited by high ATP and high citrate. Where does the citrate come from? Citric acid supplement. There. Activated by high levels of AMP and fructose. Who? Fructose 2,6-bisphosphate. Who the hell is that? I want you to underline it. He's not related. This young lady here, not related. He bashes me on the head and stop working. Just stop working, you're in my way. An unrelated molecule. F26BP is not part of glycolysis. Who's part of glycolysis? F16BP. Here are our inhibitors, ATP, citrate, here are activators, AMP and even ADP. Also this one, F26. How do they work? Let's look at the graph. Along the x-axis, increasing concentrations of fructose 6-phosphate. We increase the concentration. Let's go back here. Fructose 6-phosphate, ATP, there's my PFK1 in there. What do I make? F26BP. We notice the rate is slow for a start, increases, and then asymptotes out. That's with high ATP. I can overcome, but it's slow. Here in the green, you see low ATP. What do I need? I need a lot of ATP quickly, so get busy faster. That's why you see those rate constants change. So you see the reaction velocity with low ATP, that is, lots of AMP and ADP, get busy. Make it quickly. That's you as a student. I need lots of money. Work hard. Earn your money, otherwise I don't get to eat. Who controls the controller? Let's go back a slide. This is PFK2. Let's go back here. Here's PFK1. Go back another slide. Here's PFK1, a key regulator of glycolysis. Why? Because you're running through and activating the sugar with lots of phosphate. Once I've activated it, it kind of rolls downhill quickly. But I've got to control this controller. This controller happens to be an allosteric. Relax, busy, tense, not so busy. Relax, busy, tense. Why? Eat a meal, get busy. Don't eat a meal, get tense. So you keep changing that all the time, don't you? That's PFK1. This one's PFK2. Look at me. That's an old symbol. PFK1, allosteric, 4 subunits, relaxed tense. PFK2, one hand, another hand. One active site, a different active site. It's a bifunctional enzyme. It's going to regulate PFK1, but not directly. It's going to make F26BP. I'm going to walk you through this slowly. You need to stay with me. There's PFK2. It's a bifunctional. One hand is a kinase, the other hand is a phosphatase. It's one molecule. Kinase one end, phosphatase another. Linked together. Here it is down here. Kinase one end, phosphatase the other. Can I shut this hand down and get that one busy and vice versa? That's what this enzyme does. Well, how does it do it? Well, to do it, it needs to phosphorylate this character and it needs a protein kinase. That would be protein kinase A. What does it do? It alters the PFK2 component, puts the phosphate on. What do? You stop making fructose 2,6-phosphate. What do you make instead with the help of some water? Fructose 6-phosphate. Go on the right. When that phosphatase hand is busy and the kinase is not, you are making this molecule, F6-phosphate. You're turning fructose 2,6-bisphosphate into that molecule. If I turn that hand off and this one on, what have I got? I've got my kinase active and my phosphatase inactive. Who does that? A phosphoprotein phosphatase. Write down PP. You guys like to write down PP before? PP what? This phosphoprotein phosphatase takes the phosphate off. It makes the PFK bit active and the phosphatase inactive. It takes fructose 6-phosphate and makes in the red box F26BP. Please sit next to that. F26BP is not part of glycolysis. It's a different sugar that's kind of hanging around the outside. Like this character here. Just hanging around, causing trouble. Don't do this, don't do that. Yeah, of course, whatever. But then when she's not around, we can do whatever we want. Alright? F26BP regulates who? PFK1. When there's lots of PFK1, what do? There's F26BP, write it down. You're regulating the pathway, so a different enzyme, a bifunctional, is regulating PFK1. Why is that? Well, let's look at the pathway. Let's start up here. That's the cell membrane. There's the glucagon receptor, integral membrane protein. There's the glucagon molecule. What's glucagon doing when it's up in your blood circulation? What's it telling us? Fasting. We're in the fasting state. What do you need to do? Release glucose. What does it work to? Through a G protein. Remember we saw that last week? Stuck in the membrane, zooms along. This is the GS alpha subunit. Who does it grab onto? AC. Remember we saw that last week? Adenyl cyclase. Integral membrane protein, who did that activate? From ATP to CAMP. CAMP now can activate protein kinase A. What did you do? You now said this enzyme with kinase is phosphorylated. What are you doing? Fructose 2,6-bisphosphate is turned into that. When you're in a glucagon state, a fasted state, what are you doing? You're not making F26BP, are you? No? Comfortable with that? So it's like the regulator regulating the regulator. Now, here is our PFK1 when it's relaxed. It's busy. What's it going to do? Encourage more pyruvate production. What are you going to do? Make more ATP, aren't you? And if you're in the liver cell, you're going to make more fat or other molecules. Relaxed means the protein's open. So this character here now, remember the four subunits back here. The active site is relaxed. Substrate can get in, make lots of product. If the active site is tense, you can't get the substrate in. That means you can't make the product. So in this case, you can either get busy or you don't get busy. Unlike others, which are covalent modification, you phosphorylate them, you shut them down. Or you dephosphorylate them. Glycolysis is pretty basic and simple. You make them start with your answer. Go on this side. Yes. Head spinning there. Try and summarise some of that for you. PFKs 2. FBPAs 2 control PFK1. Who's that? What's his other acronym? Over here. What's that just got? PFK2? Yeah, that's it. PFK2 controls PFK1. We're summarising what we've just been through over the last 15 minutes or so. Regulated by various allosteric effectors as well as phosphorylation and dephosphorylation. We've seen it all semester. High levels of F6P stimulate PFKs, kinase. In the liver, phosphorylation of PFKs results in activation of FTPase, dephosphorylation of F26BP. You reduce that concentration and an inhibition of PFK1 glycolysis. In muscle, what do you get? Phosphorylation. Remember, muscle's going to store some of this too, but also work. Increasing the 2,6-BP, stimulating PFK1, glycolysis. Again, I've highlighted in red italics, F26BP. Its chemical structure is not part of glycolysis. Which molecule is? F16BP. That's the first bit before you split the carbohydrate into 2 3-carbon molecules. How do you feed glycolysis? Well, arches, jets, rooster, and what else is that? In the US, there's Wendy's, In-N-Out Burger, there's all sorts of There's my glucose 6-phosphate from the fructose down here, F16BP. There's a glyceraldehyde 3-phosphate. I've just gone simply through glycolysis, but there are other sugar moieties that come into this pathway. What you see here are things like trehalose. Hopefully you have some of that last semester, some of those sugars. Here's glucose, a nice simple one. Here's the glycogen. We take our glycogen and return to glucose. Hexakinase trapped it in there. Context. Ready for splitting. Fructose, we can do the same thing. Take fructose. Didn't we ingest fructose earlier? Yeah. Fructose is part of glycolysis. You don't have to start up here, you can start down there. Phosphorylate. What do you do over here? Well, you're taking things like mannose, another simple sugar. Hexakinase, what do you do? Isomerize it, you've got fructose. Up here, this uridine diphosphate, UDP. I highlighted that in green because we see that a few more. We saw UNP a few weeks ago. This is UDP. This is involved in a lot of pathways regulating the formation of UDP glucose, running into glucose monophosphate. This gives you an idea that things can feed into glycolysis at different levels. Those simple sugars like fructose that you absorb through your gut, through its own transporter, into the circulation can still come into glycolysis. That's a key step here. Some of these other sugar molecules will depend on what you eat. We talked about that shell, didn't we, taking lactose in their mouth, turning some of that into simple sugars, instant sweet, sour, and bread. A lot of that lactose is going to end up forming D-glucose, which is going to form by hexakinase glucose 6-phosphate. There is our child making lots of ATP, right, from lactose. Eventually, some individuals have lactose intolerant, they can't do it. See this one? You summarise some of this? Now a sigh of relief, it's almost over, isn't it? Too great. Careful, you know, there's a twist in it. Glucose, glycolysis, yields about 2,840 kilojoules per mole. That's the only number I need you to worry about. It's nowhere near as much as a simple fat like carbonate. Efficiently stored as a polymer. What's that polymer? Glycogen. Can meet the energy needs of many organisms. Put down red blood cells for a long period of time, a couple of months. It's a versatile biochemical precursor. We've seen at the beginning of this semester it can be involved in formation of amino acids, simple ones, or those amino acids formation into them. Like ketone bodies as well. It can be involved on membranes, triacylglycerols, glycoproteins, glycolipids, we started off today. It's also involved in nucleotides. How many times have I shown you a high energy carrier with a base and a sugar attached to it? There's a phosphate in it. We'll see more of it later on. So sugar's involved in those as well. Overall, you need to know this reaction. One glucose molecule, a couple of ATPs input, a couple of NAD pluses, 4 ADPs, a couple of inorganic phosphates. What did you make? 2 pyruvates. NADH, a couple of those. Protons, more ATPs, and water again. Now these could enter the electron transport chain to help with those. Depends on the cell and tissue. Please note that. Depends on the cells and tissue. Why I keep highlighting red blood cells? Mitochondria, no electron transport chain. After semester break, we'll see how our liver, and particularly our beta cell from your pancreas, manages insulin. Because you're going to have to run through glycolysis in a pancreatic beta cell to change potassium and calcium to release insulin. So glycolysis has to happen in those cells before insulin is released. And what does insulin do to you? What does insulin do to you? What does it do to your cells? encourages glucose uptake by who? By who? Who does glucose uptake?